Today we're learning about
DNA replication.
Antiparallel, pertaining to molecular orientation in which a molecule or parts of a molecule have opposing directions. Origin of replication, DNA sequence at which helicase unwinds the DNA double helix and DNA polymerase binds to initiate DNA replication.
DNA ligase, enzyme that unites broken DNA strands during replication and recombination. Semi-conservative replication, the way in which DNA is synthesized. Each of the two partner strands in a double helix acts as a template for a new partner strand. Primase, an enzyme that catalyzes the synthesis of a primer for DNA replication.
Helicase, enzyme that binds. Replication fork, a point at which a DNA molecule is replicating. DNA polymerase, primary enzyme complex involved in DNA replication.
Lagging strand. In DNA replication, the daughter strand is synthesized in discontinuous stretches. Leading strand. In DNA replication, the daughter strand is synthesized continuously. Okazaki fragments. Newly formed DNA making up the lagging strand in DNA replication. DNA ligase links to the Okazaki fragments together to give a continuous strand.
Semi-conservative replication is our replication technique. It means each strand of the original DNA acts as a template for a new strand which is added by base pairing. DNA helicase unwinds the template DNA, which is in an antiparallel, as one runs 3' to 5' and the other 5' to 3'.
DNA unwinds and replication occurs down the DNA strand, forming one replication fork. The pre-replication binding occurs when proteins in the complex recognize specific DNA sequences within the origin of replication.
For the primer, primase now synthesizes one nucleotide at a time. DNA polymerase then adds nucleotides to 3' end of the polymer and continues till sections of DNA is complete. The enzyme DNA polymerase catalyzes the addition of nucleotides as the new DNA strand grows. The newly synthesized leading strand is oriented so it can grow continuously at the 3' end as the fork opens up. The other new lagging strand is synthesized differently using Okazaki fragments because it grows in the direction away from the replication fork.
DNA polymerase then synthesizes an Okazaki fragment by adding nucleotides to the one primer until it reaches the primer of the previous fragment. Finally, the enzyme DNA ligase catalyzes the formation of the adjacent Ozaki fragments, making the lagging strand whole.